like for this purpose, if there is a crossing over that is happening non-sister chromatids here and also at this spot, then here also genes can get interchanged, rearranged. And by rearranging of the genes, we make recombinant progenies, which are not like the parents. Is it clear? This is where the variation in sexual reproduction comes. Everyone understands? Yes or no?
Yes, sir.
Yes, sir.
Then we discussed that linkage is inversely proportional to recombination, which means more linkage, less recombination. Less linkage, more recombination. Clear? And linkage depends on the physical proximity of two genes. If two genes are present very close to each other, the chances of their the chances of they getting segregated independent of each other is more or less?
Less.
Less. So if they are very close to each other, the chances that they will go together in the gamete is more. You understand, everyone?
Yes.
Yes, sir.
Perfect. So please take a screenshot of this as well for this crossing over of chromosomes. This page. Done?
Yes, sir.
Mazia, Mamuna, and Saifullah?
Yes.
Okay, Madhya has joined. Madhya, good evening.
Yes, sir.
Good evening, sir.
Madhya, you missed the last class, right?
Yes, sir, because my exam started, so I had chemistry, so I had to study.
Okay. So we did in the last class, you take the recording and please go through the class because we studied chromosome, linkage and recombination, the concept of it. Okay?
Okay, sir, I will get it.
Yes. And if you have any doubt, then what we will do is I'll accommodate you for one class. When I will do it in some other batch, I'll send you the timing, others also, and you can join that class for that particular topic if you have any doubts. Okay?
Okay, sir.
Please read that. Okay. Now, assuming that you have understood linkage and recombination, let me write down. There is a formula to calculate recombinant progenies. What percentage of progenies will be recombinant? Okay. Now and I'll also give you some questions that you will just do right now. Now write down. Formula is this. Frequency of recombination is equal to number of recombinants over total number of progenies into 100. Is it clear, everyone? Simple, right? It's simple, a percentage ratio that we are finding, correct? No complexity. Now
Yes,
let's do a question and we'll try to understand things from that question. Write down a question. Everyone is writing?
Yes, sir.
Write down a true breeding, a true breeding or pure line is the same thing. Okay, it's just another term. A true breeding plant with constricted and yellow pods, with constricted and yellow pods. When it says true breeding, for now there are two genes, remember, one gene and two gene, the pod color and the pod shape. Clear everyone?
Yes, sir.
And true breeding means, when it says a true breeding plant, it means it's pure line for both the traits. So a true breeding plant for with constricted and yellow pods was crossed with a true breeding plant, a true breeding plant with inflated and green pods. This is the parent generation. You understood? Next thing the question says, write down, continue the question. The F1 progenies, the F1 progenies were then F1 progenies were then crossed with homozygous recessive plant for both genes. Homozygous recessive plant for both genes. Okay. The result of this cross, the F1 cross, the result of F1 cross was the following. So I'm writing the result. The result was the following. So this is the result from this cross. You have to find the frequency of recombination. The cross is okay. So we are using inflated and constricted inflated and green were 445. 445. Second was constricted and yellow. 455. Inflated and yellow. 51 4 constricted and green were 49. So this is the total result. Okay, everyone. Now you have to tell me the frequency of recombination. So there are three things you have to find. First is frequency of recombination. Second, genotype of parents F1 and F2. All these two things you have to find. Can you do this? Question is in front of you. People, you also have the formula written. Are you doing it? Reem, Saifullah, Minashi?
Yes, sir.
Yes.
Yes, sir.
Yes, take your time, please do it. Every term that is written for parents, F1 and F2, you have to tell me the genotype and the frequency of recombination.
So what is the notation for constricted parts?
That I have told you, when we begin the chapter, we talked about all the 7 characters and 14 traits.
Yes,
So,
I was absent that day, right?
okay. So, tell me people, in inflated and constricted, which is dominant and which is recessive? Team,
Constricted is dominant.
constricted is dominant. You sure? All of you have NCRT with you right now? Saifullah, do you have NCRT with you right now?
1 minute, I bring it right now.
Please, everyone, open your NCRT and come to page number 70.
Constricted is recessive.
Yes. And inflated is
Dominant.
dominant. So if you are using inflated is also known as full. So if you are using any word, use the same word, okay? Capital and small. And then between yellow and green, this is the pod color, okay? Which is recessive and which is dominant.
Green is dominant.
Inflated is dominant.
Yes, inflated is dominant, and color, the pod of color, which is dominant? Green is dominant.
Green.
Okay? Yes. Now you can also page number 70 NCRT, take the help, and now can you solve it, Saifullah?
No, I mean the notation, like 40
So notation
tall
is,
necessity.
okay, let's everyone, for the sake of similarity, for inflated and constricted, we will use inflated is also known as full, okay? So capital F for inflated and small f for constricted, okay? Capital G for green and small g for yellow, okay? Now you all can solve.
Yes, sir.
Yes, please do it. I'll get some water for myself. Okay, done, people? Madiha,
Yes, sir.
done? Those who are done will write like in the chat that they are done. Okay, Mazia is done. It's just a long question to just make you think that it's very difficult or hard. It's very easy actually. Actually, all you have to do is to just make one cross, one Punnett square. That's all. I think you can also do it without Punnett square if you have practiced enough. I'm giving you a couple of more minutes to solve. Okay, Saifullah, you
Yes,
help
sir.
me? You lead me, okay? Let's try solving this.
I only did F1, sir. I have to do F2 yet.
Okay, you did F1. F2, they have all, this is F2, right? They have already told you F2. You don't have to do F2. If you pay attention, they're not asking you about F2's cross. They're asking you about the genotype of F2.
That only I'm still doing it.
Okay. So let's do it together. Let's start from the beginning, okay? Saifullah, tell me.
Okay, sir.
This is the parent generation. In the parent, there is this one. So this is parent number 1 and this is parent number 2. For parent number 1, it is true breeding plant with constricted and yellow pods. So if it is constricted, constricted is a recessive trait.
Recessive.
So what will be the genotype of parent 1?
FF.
What F? Small f, capital F.
Small f.
So small f, and
Small g, small g.
small g, small g. Clear? Everyone is clear? This is parent 1. And what is parent 2?
Capital F, and capital G, capital G.
Everyone has this answer only?
Yes, sir.
So parent is done, solved. Now you have to find for F1. When you cross these two, basically what cross you are making? Let's go a little down.
The hybrid cross.
You are making this with this. What will be all the progenies? What are the possible gametes?
Small g, small.
FG is the only type of gamete that is possible, right? Here, capital F, capital G is the only type of gamete possible, correct? Because they both are pure line. So F1 can only be capital F, capital G, small g, right?
Yes.
Everyone has this answer?
Yeah.
Yeah.
Yes,
So
sir.
our F1 is also solved. Coming to F2, you don't have to make a cross for F2. They have already told you these are the 4 type of genotypes: inflated and green pod. So what will be the phenotype of the first one? No need to make a cross. Inflated and green. Inflated means,
Capital F.
sorry,
Capital F.
but now there are 2 possible possibilities for this inflated and green. What are the 2 possibilities? Let me remove these things here. You know the notation, so I'm removing this. For inflated and green, green is dominant or recessive.
Dominant.
Dominant.
So it could be either capital F, capital G, right? Or it could be capital F, capital G, small g, right? Or it could be capital F, capital G, or it could be capital F, capital F, capital G, small g. These are the only 4 possibilities for this one. In
Yes, sir.
all these 4 cases, they are inflated and green. Yes. Tell me what are the possibilities for constricted and yellow pod. No need to make a cross here. You have to just tell all the genotypes. They are asking you all the genotypes. So for constricted and yellow, what could be all possible genotypes? How many different possible genotypes will be there? Tell me that for constricted and yellow.
Small f,
Yes.
small g.
Can there be any other?
No.
Why not? Because both are recessive traits, right?
Yes, sir.
So only this is possible. How many are possible for this? Inflated and yellow.
Capital F, small f,
Let's first take capital F,
small g,
and capital F, small g, right? Only these 2
Yes,
are
sir.
possibilities. Everyone, are you following? Yes or no?
Yes.
Yes, sir.
Adiha,
Yes, sir.
is it are you understanding?
Yes.
Yes. Now for constricted and green, capital F, capital G is one possibility. Sorry, small f, capital G, small g is another possibility, right? Any other possibility? Any other possibility?
No, sir.
No, right? So these are the possible genotypes for the F2. Now they have given you numbers of progenies that have the following to find. So everything is solved. F1, F2, P1. Clear? All we have to find in the last is frequency of recombination, for which we have a formula. To find frequency of recombination, which I'm writing in FOR, what do you have to find? Total number of recombinants. Now tell me which of these 4 is a recombinant thing. For that, we have to look at the parents. What were the parents? The parents were inflated and green, right?
Yes.
And
Yes, sir.
constricted and yellow, right? These 2 are the parent. No parent was inflated and yellow or constricted and green, right?
Yes, sir.
So these 2 are different from the parents. So they are recombinants, right? Yes or no?
Yes, sir.
Yes, sir.
Yes, everyone understands why are these two recombinants? Because they are parent type. Am I making sense, people? Say yes or no.
Yes, sir. Yes, sir.
Yes, sir.
So frequency of recombination is total number of recombinants, which is 51 plus 49 upon total number of progenies, which is 445 plus 455 plus 49 plus 51 into 100. Tell me what is the frequency of recombination? What percentage?
10%, sir.
Yes. So frequency of recombination is 10%, which means 10% of the progenies were recombinants, which means these two genes, what is the linkage between these two genes? Now, whatever is the frequency of recombination, so linkage is the maximum linkage could be 100%, right? So 100 minus FOR. So what is the linkage between these two genes?
90.
Yes. Everyone understands? The linkage is 90%. So are these genes tightly linked or loosely linked or not linked at all?
IT links.
Tightly linked.
Yes. Everyone understands what we just did?
Yes, sir.
Adiha?
Yes, sir.
So for any given combination, if they already tell you that the frequency of recombinants or recombination is 27.3%, what is the linkage between the genes? If this is FOR, what is the linkage between the genes? Hundred minus this, right? Is it very difficult?
72.7%.
The linkage between the genes is 72.7. Everyone understands this whole question? It was not difficult, right? It was
Yes, sir.
lengthy, so you thought that it's confusing and it's difficult, but it was actually very easy. Wasn't it easy?
Yes, sir.
Okay, perfect. Now, would you like to do one more question, everyone? People, would you like to do one more question? No, read. Yes or no? Shall I move forward? I hope I'm audible. Am I audible?
Yes, sir.
Check. Yes. Am I audible now?
Yes, sir.
Okay. So let's go to one more concept and then we'll do some questions together. So you understood that what is recombination, what is linkage, how it affects the law of independent assortment, and there is always a deviation from 9:3:3:1 ratio, right?
Yes, sir.
Yeah. Now write down, there is the next concept that we're going to study is, write down, polygenic inheritance. Polygenic inheritance. You know what is polygenic inheritance, people? What does the term tells you?
Multiple genes are inherited.
Polygenic inheritance it's not just multiple genes inheriting, but it's related to a character. So till now we have studied that one gene affects one character, right? Yes or no?
Yes, sir.
Case of blood group also we found that it was multi-allelic gene. It was one gene, but it has multiple alleles, like 3 alleles, right? More than 2 alleles. Polygenic inheritance is where more than 2 genes actually affect the same character. Do you understand, everyone? And this
Yes,
is
sir.
what was again uh, exception to what Mendel said. Remember, I think Reem must be remembering that in the last class we discussed why did Mendel's theory was not very popular till the 1900s, right?
Yes, sir.
Remember, one of the reason was, what were the 3 reasons? Tell me, Reem.
That he was doing mathematic applications.
For the first time, he used mathematical tools to explain biology, which biologists of that time did not accept it well. And
And then that he said that they don't blend, alleles don't blend, and that's why other,
he said that genes are discrete,
yeah.
right? They're transferred,
Yeah.
they do not blend up. Then people did not accepted it, saying that if genes are discrete, then for every kind of character, there has to be only 2 types of contrasting traits, right?
Yeah.
Like in pea plant, we can see that there is a tall pea and a short pea. There is no in between. But in humans that is not possible, right? In humans, we have height ranging from very short to very long. Yes or no?
Yeah.
And even in case of skin color, the skin color has multiple shades, like hundreds of different shades are there, ranging from very dark to very fair. So all these kind of inheritances in humans could not be explained well by Mendel's law, which is talking about in binary terms, either tall or short, one dominant, one recessive. You understand, everyone? So it's not like these genes are not following law of dominance. They are following Mendel's law, but it is polygenic. So one example is, write down. Let's first write down polygenic inheritance. Write down. Polygenic inheritance is where a character is controlled by. Polygenic inheritance is where a character is controlled by 3 or more genes together. 3 or more genes together, along with the influence of environmental factors. Along with the influence of environmental factors. Is it clear?
Sir, can you repeat?
Yeah, that is. Can someone repeat like the beginning part? Can you repeat the beginning part? Polygenic inheritance is where a character is controlled by 3 or more genes. A character is controlled by 3 or more genes together, along with the influence of environmental factors. Along with the influence of environmental factors. Is it clear?
Yes, sir.
Yes, sir.
Everyone is written. Now let's try to understand. Write down. For example, human skin color is a classic example. Human skin color. For human skin colors, let's assume for the sake of simplicity, there are more than 5 genes that affect human skin color, like which are figured out through research by now. There can be more, but let's say for the sake of simplicity, there are 3 genes, A, B, and C. Okay, or let's say there are 4 genes, A, B, C, and D. So these are 4 genes we are assuming, okay, which control human skin color. Okay, everyone. Now we also know that human skin color is also influenced by environment, correct? Yes or no? Those people
Yes, sir.
who live near equator, if you are more exposed to sunlight, then the skin color will be darker. And if you are less exposed to sunlight, then the skin color will be lighter or fair. Clear? Everyone? Now, if A, B, C, and D, if these 4 genes, you know which is the dominant skin color, dark or fair? Why does the skin gets darker? That way you can figure out yourself which is a dominant one. Why do we think there is a dark skin?
More sunlight is there.
More?
We are more exposed to sunlight.
No, that is an environmental factor. But genetically, why there is more darker? When
Melanin. Melanin is
Presence
more.
of melanin.
yes, melanin. So when someone is exposed to sunlight, what happens? Melanin is produced. But melanin is a protein, no? And protein must be producing from a gene which is transcribed into mRNA, then made into a protein. Remember? Class 11th. DNA makes RNA, RNA makes protein through ribosomes. Yes or no?
Yes, sir.
Yes. So if melanin protein is more, darker is the skin. If all the 4 genes, when they are expressed, they will produce more melanin. So expression means protein formation. So if someone's gene is, let's say this, AA, BB, CC, and DD. If someone's genotype is this is the darkest skin, right? Because all the genes are dominant and all are producing melanin a lot. So there are 1, 2, 3, 4, 5, 6, 7, 8 alleles producing melanin. So it will be the darkest skin, right? And darkest skin is the dominant one. Do you understand?
Yes, sir.
And if someone's genotype is this, AA, BB, CC, and DD, then it will be the fairest skin or the lightest skin, correct? And this will be a recessive. Fair skin is a recessive character. But between this and this, can you figure out there is a huge range? What could be all the different phenotypes? It could be AA, BB, CC, DD, right? This will be, now here, one allele is recessive, right? Rest 7 are dominant. So it will be a dark skin, but less darker than the darkest skin possible. Do you understand, everyone?
Yes, sir.
Similarly, less than that will be AA, BB, CC and DD. You understand? Are you understanding?
Yes, sir.
Then if it is AA, BB, CC and DD, it's going to be even less. Yes or no?
Yes, sir.
And finally, if it is the pairs are AA, BB, CC, DD. When you say two copies are homozygous recessive, only two copies then are producing melanin, right? This will be a intermediate skin. And when we will reach here, all four copies are homozygous recessive, it will be the fairest skin. You understand? Everyone understands, right?
Yes, sir.
So this kind of inheritance where multiple genes give a gradation in the character is called polygenic inheritance. So in case of skin, write down. In a polygenic inheritance, the number of each type of allele,
Can you repeat from polygenic inheritance?
yes, in polygenic inheritance, the number of each type of alleles in the genotype will determine the will determine the nature of the character in an individual, the nature or gradation of the character. Same is for human height. For height also there are multiple genes, around 6 genes in humans. So that's why when all these genes are together and all are dominant, the person is very tall. But all chances of all being very dominant is very rare, right? And the chances of all being homozygous recessive is also very rare. 9:3:3:1 is that all recessive, right? But any gradation in between will give rise to different heights. You understand? So another example
Yes,
is
sir.
human height. Is polygenic inheritance clear?
Yes, sir.
Very good. And this release is also governed by the environmental factor. If someone who is born with this genotype but is never exposed to sunlight, you are born with this. So when you are born, there is melanin, but over time, if you are not at all exposed to sunlight, suppose there is a baby born to an African couple. Of course, they will be having dark skin because to survive there, you need dark skin. But right after birth, you send that baby to some very cold region to just grow, never exposed to sunlight. Then the skin tone will be less darker than the parents, right? Not because of genetics, but also because of the environmental influence. Yes or no?
Yes, sir.
And if a kid is born to, let's say, a Kashmiri couple or a European couple, of course they will be fair, right? Because they live in cold places. But just after birth, if you send the kid to Africa to just grow, over time it will start producing more melanin, right?
Yes, sir.
Yes, sir.
Yeah. And the skin tone will become dark. It happens with you also, right? When you go into sun, with the same genetic makeup that you have, your genes do not change when you go into sun. Yes, but you get tanned, right? Yes or no?
Yes, sir.
So you understand, in polygenic inheritance, there is environmental factor also which plays a role. Same is for height. With the same set of genes, if someone is actively doing exercises or postures or the kind of activities that help in height, lengthening of the height, the person can have a longer height, right? It is also seen in twins. Have you seen identical twins? Sometimes one identical, they both at the time of birth are identical twins, but as they grow, due to environmental factors, one can become more obese and one can become less obese. If one likes to eat lots of junk food, other does not like junk food at all. So just by their behavior and the lifestyle and the environmental factors, one can become obese, one can become lean, right? One goes and settles in Africa, other goes and settles in Europe. There will be change in the melanin secretion also. Yes.
Yes.
One is
Yes,
very
sir.
active, always likes to do gymnastics. Another is very lethargic, not playing games. Then height can also be different, right? Yes or no?
Yes, sir.
So which proves that even with the same genetic makeup, due to environmental factors, the genes which have polygenic inheritance, especially those genes, can vary in a graded manner. Clear, everyone?
Yes, sir.
Okay. Now let's come to another concept which is called, which is different from this, and it's called pleiotropy. Pleio. So what is pleiotropy? Any idea? Till now, we studied two things. One gene affecting one phenotype. Yes. Yep. We also saw multiple genes affecting one phenotype. Yes.
Yes.
Pleiotropy is opposite of it. One gene affecting multiple phenotypes. You understand? So write down. Pleiotropy is where a single gene can exhibit or influence. Single gene can exhibit or influence multiple phenotypic expressions. Do you think this is also not following the law of Mendel? Does not fit in either of the three laws, correct?
Yes, sir.
Yeah. Write down, such a gene is called pleiotropic gene. Gene is called pleiotropic gene. One example which is famous and it is asked in entrance is write down. There's a disease known as phenylketonuria. Phenylketonuria. So write down. Phenylketonuria, which happens in humans. Phenylketonuria, which happens in human, a disease which happens in human. Write down, this disease is caused by mutation in a single gene. Caused by mutation in a single gene. Which single gene, and what does this gene do? That codes for this gene, codes for the enzyme phenylalanine hydroxylase, that codes for the enzyme. So there is one single gene that codes for this enzyme. The enzyme is phenylalanine hydroxylase. You do not have to remember the name of the enzyme, but you know that it is a single gene mutation. So it is a single gene. It is a case of a single gene mutation, right? Everyone understands? There is only one
Yes,
gene
sir.
that is mutating. So it is easy to understand if there is one gene that is mutating. For example, if in a Mendel's cross, let's say the gene for height got mutated. So what will be affected in that plant? Just the height, right?
Yes,
or
sir.
no? Will the flower color, the pod shape will be affected?
No.
No. Here also there is a single gene mutation which causes a disease called phenylketonuria. And that mutation happens in the gene that codes for the enzyme phenylalanine hydroxylase. So you will assume, you will just think that, okay, one enzyme is getting faulty. So something in the body will get affected. One thing will get affected, right? But instead, in this disease, many aspects gets affected. For example, write down the manifestation of the disease. So there's a single gene mutation, but the manifestation of the disease is characterized by, so the phenotype, the manifestation of the disease, manifestation matlab how the disease progresses. Is characterized by, if someone's insulin is mutated, let me ask you this. You know diabetes? Diabetes as a disease, you all know.
Yes, sir.
Yes, sir.
What happens in diabetes? What happens in diabetes? You said yes. You know about diabetes. What is diabetes?
The body stops producing the required amount of insulin.
Yes, body cannot produce functional insulin, or the insulin gets mutated, the gene got mutated. So if the insulin gene got mutated, someone will have a by birth diabetes. In that case, the body will not be able to produce a functional insulin, right? But because of that, diabetes will happen, right? Yes or no?
Yes, sir.
One disease, blood glucose level will always keep rising. So you have to give insulin injections from outside to manage it. Simple thing. Here also one gene got mutated, but in this disease, what all things happen? The first thing, the first phenotype that happens is mental retardation. Now, mental retardation is related to brain. Yes or no? In phenylketonuria, the patient has mental retardation, cannot read, learn, or cognate properly. The brain is not developed, right? The second
Yes,
thing
sir.
that happens is reduction in hair fall. Now, do you think reduction in hair and brain retardation are two very, very different things?
Yes.
And also in this, reduction of hair pigmentation, which means hair starts to get like less and whitish, grayish. And also there is reduction in skin pigmentation, skin color. Now I already told you skin pigmentation or skin color is affected by some other gene, right? Multiple genes are there. But when you see this disease, don't you think that there is lots of unrelated things happening, but due to a same single gene mutation?
Yes, sir.
Which clearly tells you that this gene, phenylalanine hydroxylase, was playing a role in brain development, was also playing a role in hair pigmentation, and was also playing a role in skin pigmentation. Yes or no?
Yes, sir.
So a single gene is affecting multiple phenotypes. Is it clear?
Yes, sir.
This is known as pleiotropy. Write it down, everyone. Let me know when you're done, people.
Done, sir.
Done, everyone?
Yes.
Yes? Okay. So now, the next major, before going to the next topic, which is a big topic in itself, sex determination in multiple organisms. You already know about sex determination in humans, right? Can someone brief me about how sex is determined in humans quickly? I will ask, let's go to Madhya. Madhya, are you there?
Yes, sir.
Can you tell me about sex determination in humans? How is the sex determined in humans?
Okay, so
Tell
the
me everything
female
that you know about it.
contains the X chromosome and the male contains two different types of chromosomes, X and Y. So like when the female gets an X chromosome from the male, it will be a girl child, and when the female gets a Y chromosome from male, it will be a boy. So like XX is girl and XY is boy.
Okay. And is there any other thing that can affect the sex of humans?
Mutation in the gene, the male gene.
Which gene? Is there any one gene that decides the, that is what I'm asking. Do you think there is one single gene that decides the sex, and if there is a mutation in that gene, the sex will get disruptive? Or
I don't think so. There is one gene. The chromosome is one, but and gene.
yes, the chromosome is one, but the gene is not one, right?
Yes, I guess.
Yes, so you guessed right. Do you know, have you heard of any other ways of sex determination, people? I'm sure that you have heard about and read about it in junior classes. You just have to remember. Do you know some organisms, sex is determined not by genetics, but by the environment? For, do you know it or you don't know it? First, tell me that. Yes or no? Do you know about reptiles? Say yes or no. Do you know about reptiles? If their eggs are kept at a lower temperature versus a higher temperature, there will be difference in the gender. You know about that? People, say yes or no. You don't know about this. Okay, cool. So let's do one thing. Before going to sex determination, till this part? Is there any confusion in any of the topics till this part? Because I want to discuss some previous year's questions to see how well you are learning and how well you can solve. Do you have any doubts? This is a time to ask. Otherwise, I will go for some questions. Any doubts? Minashi, Reem,
No, sir.
Mazia?
No, sir.
No? Okay. So let me stop this share for some time. Some questions. Okay. Yes. Can you see the screen, people, that I'm sharing with you? Is it visible to all of you? People, is the screen?
Yes, sir.
Yep. So I'll go with Reem. Reem, can you? So this is question number 2. Can you please read question number 2 and read the options for all of us? It was asked in 2017 in the NEET. So
Okay,
all of you will do one thing. You will write the answer in the chat just to me. Just the option A, B, C, or D. Okay. Reem, question number 2.
The genotypes of a husband and wife are IA, IB, and IAI, small I. Among the blood types of the children, how many different genotypes and phenotypes are possible? Option A, 3 genotypes and 4 phenotypes. Option B, 4 genotypes and 3 phenotypes. Option C, 4 genotypes and 4 phenotypes. And option D, 3 genotypes and 3 phenotypes.
Yes, it has been asked recently. You all understood that small I is the allele which codes for O,
Yes, sir.
no sugar. Yes, everyone? Now tell me the answer quickly. Time starts now. Everyone, write the answer in the chat quickly. Just the option. I'm getting some interesting answers in the chat. That's good. Okay. Okay. Mamuna and Mazia.
Yes, sir.
So I'm waiting for your answer. You understood the question, right? There was no problem with the question. Do you have any problem with the question? Mamuna, are you solving it? Or are you stuck somewhere?
Sir, I'm doing.
Okay. And Mazia also. Mazia, you're doing? Okay. Try to be fast, but in the beginning, I will allow if you are slow, but you are correct. It's okay. It's a good thing. Over time, you have to be faster. All you have to do is to make a F1 simple cross, right? And it is not polygenic. It is just codominance, right? In blood group, there is codominance. Yes. Okay. Ramana,
Yes, sir.
Mamuna. Talking to Mamuna. Saifullah, I have received your answer. I'm yet to receive Mamuna's answer.
Sir, 1 minute, task start.
Sorry? Come again, please.
Sir, I'm sending the answer. 1 second.
Yeah. Okay. And with questions, I would like you to be faster. Yes. So almost all of you have given the right answer. So let me tell you, let me discuss the right answer. So I will discuss only the right answer and then you understand. The right answer is B, right? There will be 4 genotypes, but only 3 phenotypes. Yes or no?
Yes, sir.
Yes, Sefla, would you like to tell what are the 4 genotypes? Just the genotype, like IA, IB, things like that.
Yes, sir.
Yes.
IA,
Yes.
IB,
Yes.
IAI, and IBI.
Yes. So these will be the 4 genotypes, right? But
Yes.
what will be the 3 phenotypes? Blood group.
A positive, AB, and B.
A positive, no, it's just A,
Just say yes.
AB, and B,
And B.
right? Yes,
Yes.
perfect. So 4 genotypes and 3 phenotypes. Do you understand? Madhya was right. Mazia, do you understand Mazia? There will be only 3 phenotypes. Yes. Okay, perfect. There are certain things that I have not taught you. I'll teach you this. Let's go to one more question. Question number 10. Saifullah, would you like to read question number 10?
Yes, sir. A tall true breeding garden pea plant is crossed with a dwarf true breeding garden pea plant. When the F1 plants were selfed, the resulting genotype were in the ratio of. Should I read the options also?
Yes. So the options are in front of you. Everyone understood the question? Now tell me the answer. Don't read the option. Write the answer similarly like in the chat. It's a very simple question. If you can figure out the genotype of the parents correctly, then it's a sugar candy. I have not received any answer. Why? Is it difficult question? They're just twisting the question. You're just tricking your mind. You're thinking that it's a difficult question. It's a very easy question. See answers now. Madhya, read the question once again. Just read it. I know you know the answer. Just read the question once again. Madhya, just pay attention to what ratio it is asking. What it is asking you to tell. Mamuna, Mazia, Reem. Reem, are you there?
Yes, sir. I'm still doing.
What are you doing in this? There's nothing to do, people. It's just a simple classic case. Let me, so I don't know. Should I allow you time to complete or should I tell you? Madiha made some adjustments. She realized that it is asking genotype and not phenotype, but still there is an issue. Okay, see, you have to do nothing. The question is asking, yes, I'll now tell you the right answer. The correct answer is option C. 1:2:1, because the parents are capital T and the other parent is small t. Yes, everyone? Everyone
Yes, sir.
understands? Madiha, do you understand? The one parent is capital T, the other is small t. So when they will be crossed, all the progenies in F1 will be capital T, small t, hybrid tall. Yes? So there will be no ratio. Yes, Madiha?
Yes, sir.
If you self cross capital T, small t, there will be one homozygous tall, capital T. Two will be heterozygous tall, capital T, small t, and one will be dwarf, small t. The classic question without any exception. Answer will be C. You just got confused between tall homozygous and tall heterozygous. Yes? 1:2:1. C option will be the right answer. Everyone understands why C is the right option. These are the
Yes, sir.
type of questions that come in entrance. Okay. Now, Reem, I'll ask you to read question 17. You can just answer. Read and answer, Reem. Question 17.
A pleiotropic gene. Should I read the options?
Just tell me the answer. What does a pleiotropic gene is or it does?
Option B.
Yes, it's simple, right? It's option B. You understand? Yes, Madiha, correct. Now, Reem, you have to read question number 12 and read it carefully. Also read the options. And everyone, I want right answer. So please think. Question number 12, Reem.
In a test cross involving F1 dihybrid flies, more parental type offspring were produced than the recombinant type offspring. This indicates, I should read the options.
Yes.
Option A, the two genes are linked and present on the same chromosome. Option B, both the characters are controlled by more than one gene. Option C, the two genes are located on two different chromosomes. And option D, chromosomes fail to separate during meiosis.
Yes. Which is the answer? It is just testing your linkage and recombination concept, right? Reem,
Yeah.
tell me the answer, people in the chat quickly. I received the first answer. Very good. Try to be fast now. Adhiya, you have sent two answers. Which one should I consider?
Sir, the one which I sent just now.
Later one.
Yes.
Quick, everyone. These type of questions you should not spend more time on. You just have to apply the mind, save some time for other questions. You have to be faster with time. Entrance is all about race against time. You know everything. If I give you the same question paper that NEET gives you every year and allow you to solve it in one full day, all of you will top the exam because you have a full day to think. But the problem is you just have around 180 questions and 180 minutes. So 1 minute, one question. That's where it makes a difference. You have to be fast and you have to be correct. And you have to also decide whether to attempt or not because there is negative marking. So these things makes an entrance difficult. Yes, I've received answers. All answers are correct till now. Very good. Madhya, Minashi and Reem. Correct. Others, quick. Now from next question onwards, I'm going to start a timer. Right, Saifullah. But why are you so late in answering? Mamuna, you have to be fast, girl. I expect from you. Mamuna and Maziha. Some pressure handling is good in class. Mamuna and Mazia, you have 10 seconds, both of you. Give me an answer. Am I audible, Mamuna?
Yes.
Yes. Are you not able to register the answer to me, or you're not answering? Then we'll also discuss answer. Okay, so let's discuss the answer. In a test cross involving F1 dihybrid flies, more parental type options. Now, this first line is just to confuse you. In any given cross of the world, whatever name you give it to it, test cross, back cross, this cross, that cross, dihybrid cross, monohybrid cross, what matters is more parental type offsprings were produced than the recombinant type offsprings. This is the only sentence that makes sense here and is important for the answer. If more parental type offsprings were produced than the recombinant types, it clearly means that the genes are linked, right? That's why recombination is not happening enough. Is it clear, everyone?
Yes, sir.
Yes. So if you just get confused by the first line and try to understand, oh, what is a test cross, F1 dihybrid, what is it talking about? No, it's saying more parental type offsprings are produced than the recombinant types, which means there is less recombination and more linkage. Just the concept of linkage inversely proportional to recombination. So the correct answer is A. The two genes are linked and are present on the same chromosome. Both the characters are controlled by more than one gene. No, that is polygenic inheritance. Polygenic inheritance has nothing to do with parental type or recombinant offsprings, because there will always be gradation. The two genes are located on two different chromosomes. If they are located on two different chromosomes, parental type offsprings cannot produce, right, more, because they are two different chromosomes. So obviously they are going to segregate. Chromosomes fail to separate during meiosis. No, that's also not the answer. Answer A. Okay. Understood? Namuna, Mazia, Saifullah?
Yes.
Yes, sir.
Yes? Okay, perfect. So now I'll put a 45-second bar. Okay, let me start the timer. And I'll choose a question. Question number 14, and it will be read by Mamuna. Mamuna, please read question number 14 and also the options.
A gene showing a codominance has option A, alleles that are recessive to each other. Option B, both alleles independently expressed in the heterozygote. Option C, one allele dominant to one another on the other. Option D, alleles tightly linked on the same chromosome.
Yes, time starts now. 45 seconds. Send me the answer. Quick. Yes, now it's good. Getting answers. Yes, yes, yes. Okay, so all what was needed was for me to put the timer. Okay. And very good. The answer is option B, both alleles independently expressed in a heterozygote condition. Okay. Question number 22, Reem, please read. And also the option. The moment you will finish, the timer will start. Question number 22.
Multiple alleles are present. Option A at the same locus of the chromosome. Option B on non-sister chromatids. Option C on different chromosomes, and option D at different loci on the same chromosome.
Time starts now. Multiple alleles. What are alleles? They are the different versions of the same gene. Okay. There is still 30 seconds left. Minashi, did you change your answer? Okay. Saifullah, you also change your answer?
Yes, sir.
Amuna,
Sir is sent.
and
Yeah, for the 14th question, right?
no, for the 22nd question. Question number 22.
Okay.
Saifullah, let me tell you one thing. You were correct in the first instant. I don't know why you got confused. Okay.
Okay, sir.
Correct. So everyone's answer is there. Right answer is option A. Multiple alleles. Alleles are the different versions of the same gene. So if it is the same gene, it has to be present at the same locus on the chromosome. So if there is a gene for eye color present on chromosome number 1, so it will be present at the exactly same location on chromosome number 1 from the mother as it will be on the chromosome number 1 from the father. This is because when crossing over will happen, then only you can make proper recombinants. Okay, so alleles cannot be present at different locations. You understand? Madiha, Saifullah.
Yes, sir.
Yes, sir.
Minashi is the one who changed the answer to correct. Minashi first answered the wrong one, then she corrected. Saifullah did the exact opposite. Okay, good. But I'm glad that you all are like big pace. I have some important questions again. Yes, 23. Question number 23. Madiha, will you please read?
Yes, sir. A man with blood group A marries a woman with blood group B. What are all the possible blood groups of their offsprings?
Yes. Quick, 45 seconds you have. Madiha, pay attention to the fact. Are they telling you the genotype of the man and the woman? No. Then why are you assuming that the genotype will be what you are assuming and hence giving me the answer, which is not the correct one? So please don't assume. They have not given in the question. It's open for possibilities. A blood group. Yes, right. I have received answers. All the answers. Yes. Okay. So let me. Yes. Now I received all the answers. The right option is A. They are not telling you whether the blood group is heterozygous or homozygous. So you don't have to assume IA all the time. It can be IA or IA or IAIO also, right, Madhya?
Yes, sir.
For B also it can be IB or IBIO. In which case, whichever it is, all the 4 blood groups are possible. Just a trick question. Very easy, but just to trick you. Okay? Everyone understands? Now, I'll discuss one thing here. How many pairs of contrasting characters, question number 24, in pea plant were studied by Mendel in his experiment? So the answer is 7, okay? You know the answer already, it's 7. But there was another question, which is in somewhere in here also, I've seen it. They just twisted the question to ask, how many varieties of pea plant were studied by Mendel? So if they write down this question, if they ask you how many pairs of contrasting characters were studied, the answer is 7. 7 pair of contrasting characters. But if the question is how many varieties of pea plant, then it is 14. 7 into 2. So each tall is one variety, short is one variety. You understand?
Yes, sir.
Yes, variety means variations. So total 14 varieties and 7 contrasting characters. Just remember that. Did everyone understood what kind of questions comes from the topic that we have covered till now?
Yes, sir.
Yes, sir.
Yes. So please revise and look for questions like this from previous year's papers and sources. And in the next class, when you will come, we will finish other concepts and do more questions. Okay.